Okay, so today I'm going to talk about Beyond the Lab Notebook, assessments for course-based undergraduate research experiences, or as I will refer to them as cures. So what are the dimensions of a cure? If you look at the images here, it involves the use of scientific practices, collaboration amongst students, iteration, discovery, and broadly relevant or important work. So I'm going to go through of each of these categories and give everyone a sense of what that looks like in terms of traditional lab learning, inquiry-based learning, and then cures. So if we think about the use of scientific practices, we want students to be asking questions, building and evaluating models, proposing hypotheses, designing experiments, selecting their methods, using tools of science and whatever field that might be in, gathering and analyzing data, identifying variation, and navigating the messiness of real-world data. We also want them to develop and critique interpretations and arguments, and then communicate their findings. So if we look along the top bar, this table shows the use of scientific practices, and then kind of breaks that out into students engage in and the study design and methods are. And then it goes across in the columns, traditional, inquiry-based, cures, and internships. And so what we see in traditional lab learning, that there's few scientific practices because they're often given a protocol, and it's more of, sorry, someone wants in, more of a learning of the protocol rather than understanding of it. And the study design and methods are obviously instructor driven. If we move into inquiry, there are multiple scientific practices that are student driven. And if we look at cures and internships, there's multiple scientific practices involved in undertaking research, and they can be student or instructor driven. If we look at discovery and what that kind of encompasses, we have the purpose of the investigation is instructor defined in traditional learning, student defined in inquiry based, and in cures and internships or independent projects, it can be student or instructor defined. The outcome is really a really important aspect of cures because it has to be unknown. Inherent in doing research is that the findings are novel. And so this is where we see a pretty big difference between traditional and inquiry-based learning compared to cures. If we think about the broader relevance of students' work, in traditional and inquiry-based learning, it's usually limited to the course. It's, an example of some scientific concept, and it rarely presents opportunities for action or communication broadly. However, with cures, the relevance should extend beyond the course, given that the question and the data will be novel. And so it often presents opportunities for students to present their work, maybe connect to different societal, environmental, or community issues, and potentially opportunities for collaboration across institutions or disciplines. When we look at collaboration, in terms of traditional and inquiry-based lab work, it often occurs between students in a course. However, with a cure, we have it among students, teaching assistants, and instructors in the course. And the instructor's role is generally guidance and mentorship rather than instruction, which is another key defining piece of a cure. In terms of iteration, obviously part of iteration is that there's a risk of generating messy data, and that's inherent in doing novel scientific research. And of course, we don't ever only do an experiment once. when we're trying to understand something. So we have iteration built into the process where they might try to achieve an N of 3 or something like that. So if we look overall at this table, what we can see is that cures have the most similarity to internships or independent projects. So that's great. Hopefully everyone has a good understanding of cures now, but what are the benefits of cures? So cures have been around for over a decade. There's been a lot of work in developing them and kind of, understanding the benefits of them. And this is a table from a publication where they showed that students benefit in several aspects. They increase their content knowledge, their analytical skills, their self-efficacy. They have external validation from a science community. They persist in science and really understand those skills. And they have potentially career clarification because they get to experience real research. In the classroom, I've noticed, as well as others, increased project ownership, their increased communication skills, definitely a huge motivation with that ownership, collaboration, and importantly, tolerance for obstacles, because inherent in doing experiments where the outcome is unknown is the fact that it's likely not going to go the way you plan, and you have to troubleshoot and really rethink what you're doing. Increased sense of belonging to a larger community. Obviously, if they're generating a novel research question based on scientific literature, then they're actually seeing how that question fits into that scientific field. And with that is an enhanced science identity and positive interaction with their peers. If we look at interaction with faculty and mentorship, cures increase the ability for students to gain that. And inherent in doing research is that they have an enhanced understanding of the nature of science and the development of self-authorship. For faculty, I think, in my experience, the unexpected data provides a greater opportunity to teach the true nature of scientific process. And that really has enriched my teaching and the student engagement in my courses. It expands my mentoring capacity. If I'm, undertaking independent project or honor students, I can take maybe 2 a semester. If I run a course of a cure, I can have 14 students in that one semester. It also allows the integration of teaching and research. If we think about EDI principles, cures actually are interact with the EDI principles quite well and support making research more accessible to students through course integration. They don't have to know how to find a professor or, have the initiative to look into research projects and find a professor to work with. They just have to register for a course so they can gain that research experience. It also breaks down barriers for students who might not know that they need research or know anything about research or why it might be useful for professional programs or other things. So it just breaks down a lot of barriers. It encourages collaboration and diverse teams, and the students see themselves as scientists. So I think that's really important. Okay, so if we look at the 4-step cure pedagogical framework, this kind of identifies the pieces that need to exist within a cure. And so obviously, you've all carried out research projects, and this is nothing new to you. So it's pretty much the same. Learn the technique, understand the literature and find a question, design an experiment, develop the protocols, carry out the experiments, collect and analyze the data, and then interpret data and communicate results. So I've been teaching in the the Advanced Cell and Molecular Biology lab course for over a decade now. And within those 2 semesters that span the 4th year of the cell and molecular biology concentration in our biology degree, they are 6-hour lab courses. And we basically were doing this traditional assessment. So we didn't even really know that we were doing a cure. But when I really started to, work through this process and encountered some of the issues, I really started to see, number one, it was a cure. And number two, that this is most likely where people might start when they're developing a cure. Especially in upper level 1. So you start with your research proposal and get the students to understand where their question fits in with the literature. And then they write in their lab notebook as they design their experiments and carry out their experiments. And then in the end, they submit a manuscript or make a poster of their data. Well, I found that there was significant challenges with the assessments. In terms of the lab notebook, the students often would come unprepared to the lab. Now, remember, in this process of developing protocols and carrying out experiments, I'm not giving them the protocol. They're expected to come to lab ready to do the experiment. And what I found is that there was a very big leap from being given a protocol in a traditional lab and coming to lab and doing it compared to, being given general protocols, but having to figure out all the details. So what I found is that the students would come unprepared. They wouldn't have the math figured out. They'd make mistakes. They'd waste reagents and time. In red, the most significant challenge for me was how time consuming it was to grade a lab notebook. Students often, if they're not given very, very clear guided instructions of how to structure their lab notebook, which is kind of opposite to what we want from a lab notebook, everyone has different stuff in their lab notebook. It's not consistent. And then you go through and you write all the comments and you correct everything. And often the students don't even go back to read it. So I think the time that it took for me to go through those lab notebooks every week or 2 weeks really forced me to rethink how I could be giving them better feedback and preventing the wastefulness and the unpreparedness. I also noticed, because they didn't have a lot of formative feedback about their data or their understanding of how to analyze the data, when they submitted their final manuscripts, there was often misconception or errors that at the end of the semester was now really too late to correct. And again, we know that students often won't read the comments at the very end of the semester. And often the manuscript has a substantial weighting of the overall grade, which I think is challenging as well without all that formative feedback. So if we look at this slide, on the top of the slide is the cure pedagogy, just shown different than the last slide, but the same content. And then the traditional assessments that are often used when we start this process. And then what I did with deconstructing the lab notebook. So what you can see here is I developed 2 different types of assessments: experimental plans and data submissions, and kind of shifted the weighting of this deconstructed lab notebook scenario to the traditional assessments. So in our course, when it ran previously, the lab notebook would be 10 to 20%. And then a lot of the grade was on these final communication pieces. In the deconstructed lab notebook, the experimental plans and the data submissions are about 50% of the grade. And that's really trying to uh, give the students the impression that that process is important, and putting the grades to the fact that the process is as important or more important than the outcome. So what do these experimental plans and data submissions look like? I'm going to talk and give you some examples of what I've done. So in this course is the molecular biology component of it. I'm going to show you exactly how I developed it, because I believe it's easy to develop for any experiment that you might want to do. But basically what we have here is the first 5 weeks of the course. And you can see that the experimental plans are due immediately. They have to start working on their like setting up their protocol. And because the students find them really challenging initially, I have an incremental increase in the weighting of them, so that they're low stakes to begin with as they learn the process, and then they can build up to having them worth more. So these are the instructions that are given to students. So obviously I will post the protocols that they need and any specific details that they wouldn't know. So for example, in this course, they're growing muscle cells. They don't know, what dish we're growing in and things like that. So I do provide them with information that they couldn't know at that level. But I do expect them, when they write their experimental plan, to go through and tell me the protocol with all the details. So, for example, when we take out a protocol, it might say, add X times 3 volume of your cells, or, different things that you have to go and calculate. So they actually have to go and be completely prepared. They have to identify the purpose of each step, what the reagents are and what it's being used for in the experiment, and then any controls that they might need and what the purpose of those controls serve. Now, obviously, I understand that this is very specific to a molecular biology course, but we have used it in several iterations of different courses. And, obviously, you just have to fine tune it to your experiment. The requirement is that they need to demonstrate thoughtfulness in the design of their experiment. So if they need to prepare a reagent and it says you need 300 microliters of this reagent per sample, they need to know how many samples they're going to have. So they need to know how much reagent they need to prepare. All of these pre-thinking exactly what we would all do, but forcing them to do it before they come to lab so that they can actually get the work done in the lab time. They also have to demonstrate that they understand how they'll interpret the results. So here's an example of a student's experimental plan. On the left is their exact submission. And on the right is my marking breakdown. And I hope to convince you that this can make marking really, really simple. So basically what I do is I have a spreadsheet, like an Excel file, and I have the required component, and each of them are 1 mark. No half marks, it's 1 or 0. And they get used to it real fast. So what is the purpose? Did they write the correct purpose? If they did, they get 1 mark. In this case, the student did. They said why we need to determine an optimal reference gene for this quantitative PCR experiment that we want to run. And then within the protocol of isolating RNA and validating it and performing reverse transcription, I identify the things that they would have to think about, things that we would tell them in a traditional lab, but things that they would need to think about in designing their own experiments. So For example, in this experiment, we had a myoblast. These are muscle cells, a myoblast and a myotube sample, and they needed to identify that they would validate their RNA with spec and the ratios and what have you. And it's literally, did they say it or not? Check. So it's really super fast. Um, and then because we were doing a lot of validation experiments in this semester, they had to identify that they would do this validation experiment. So it's literally not nitpicking through every single detail, but do they have those key pieces that when they come to lab, if they haven't thought of, they're going to have to sit there for half an hour trying to figure it out and waste that lab time, so then they can't get the work done, and then the whole timeline is put back. So again, this is just a continuation of the student example and the things that I was looking for. So I think, you know, if you're considering something like this, the first semester of building these like marking breakdown rubrics is a little bit time consuming, but after it's done, it's like super fast and easy, and you can modify them based on the project. As I can talk about that a little bit more. And then did they think about the analysis and what they're going to get out of this experiment? So there is guidance, but there's also the requirement for them to look at this experiment in a completely different way than in a traditional lab. So the benefits of this experimental plan is that students come prepared, and basically in my course outline schedule, all the experimental plans are submitted and marked by me before they start the experiment. So any misconceptions or errors I can fix before the experiment begins, not after, when we're trying to troubleshoot what went wrong. And with this marking breakdown with 1 or nothing, you're just looking for those key things. It's really quick to grade. Another thing I've done is I've actually had them, depending on the experimental plan and how critical it is or how many errors students made, I would get them to share it with another student and go through it at the beginning of lab, and they could grade it in like a Google Doc, for example. So I've run this since 2019, and I've asked students to give me their perspective on experimental plans. And so I'm just going to tell you a little bit about things that they've told me. So they said they feel more prepared for lab experiments. They said they hold a deeper understanding of the protocol and why they're actually doing it. They said they derive more meaning from the results upon completion. They understand the experiences of a researcher to a higher degree, and they feel more secure and confident in the 3rd to 4th year lab transition. So that's actually a student that we did this with in a 3rd year lab course. They helped me to understand what we were doing in the lab and how it related to previous and future labs that were occurring. It made the labs flow very nicely. I know this is long, but this one really hit me because I'll read it to you. So I just wanted to say that I think going over the procedure before the lab is one of the most helpful things in a lab-based course ever. I don't mean just simply reading the procedure, but actually understanding the processes, tracing products and transformations, and doing the math beforehand. I try to get as much of the math as I can done before the lab, as I tend to panic when trying to do it on the spot in a time-sensitive environment. And that really struck me because it was something that I hadn't even thought of in the design of these experimental plans. But I can actually see it in many students that they are quite happy to not have to, you know, be flustered and trying to think about these complex things on the spot, but rather in their own time. Okay, so I talked about deconstructing the lab notebook into experimental plans. Now I'm going to talk a little bit about the data submissions. So again, general instructions to students for every single data submission. They need an explanation of the purpose of the data in the overall project. Obviously, many projects are going to scaffold varying experiments, whether it's multiple different kinds of experiments or a whole series of things that you need to do before you can do an experiment, such as the case I'm describing, where they had to grow the cells, they had to isolate the RNA, validate it, synthesize cDNA, and then do their qPCR. So they need to have the raw data depicted in a figure or a table, a figure caption, key results in point form, and clear interpretation of the results. So I kind of see this as like a preliminary figure. So here's an example. So this is in an Excel spreadsheet. They have their data, they have their graph, they have their figure caption, and then the key findings of that figure. So again, I'm not looking for polished writing. I'm just looking, do they actually have their data presented correctly? Have they thought about what their data is showing them? And can they say something about the data? So for the data submission, I have found that the students actually, it ensures the students are interpreting and presenting their data correctly, but it also allows formative feedback before they put their figures into their manuscript. And it also gives them an outline of the written results. So one of the things that I found with final manuscripts is that it comes at the end of the semester, the students are pounded with all sorts of things in their classes, and they just don't have enough time to sit and think about their data and really present it in a meaningful way. But if they've already made all their figures and an outline of the results, all they have to do now is either correct it if they've made some errors, but just start building their manuscript over the semester. So like I said, the overall benefits, they come prepared, they have a solid understanding. I should mention with the data submissions, obviously, one of the problems that I've seen is student anxiety around, what if I don't get the data that I need? Am I going to fail if my data doesn't work or my experiment doesn't work? And so with this data submission, it is a new thing that I added, but what I also say is everything is data. And even if you don't get the data that you need, for example, if your optimal reference gene didn't work, that's still data. You can still talk about it. So there's always the opportunity to submit their data, whether it's good or bad. It's still data that they can talk about. And if they don't get the outcome that they wanted, they can talk about why things might not have worked and what they would do differently next time. I think as well, one of the issues with collaboration on research projects and collaboration on assessments is that we don't want to have too much of the grade on group work. So in this model, the experimental plans and the data submissions are individual, and it's over 50% of the work or the grade. So then they have the ability to do some collaborations on assessments and not have a huge part of their grade part of that. So this just shows that. So the experimental plans and data submissions are completed individually, and they're worth a substantial amount of the grade. I alluded to building this manuscript, and I think it has worked really well for myself as an instructor and for the students. So the experimental plan really can become the basis of the written methods, which is the method section of the manuscript. All of their data submissions will then become a figure, and the figure captions and the outline of, oh, I have a spelling error there, of the written results. And so when we come to submission of that final manuscript, all they need to do is fix up that introduction and write a discussion. So the marking load at the end is really about that interpretation of the data rather than all the things that they did throughout the semester. So some final thoughts, and then I'll open it up for a discussion. So with the experimental plans, the purpose is to have that critical thinking and design outside of the lab, so that the students come prepared and their misconceptions are corrected before they do the work. It's easy to peer grade in class, or do it outside just before they start the experiment. The data submissions obviously allow the students to build their skills in figure creation and really force them to think about the data early on. Obviously, in real research, we base every experiment on the one before, and so building that in really forces them to think about that. And of course, for us, it makes marking the manuscripts a lot easier in the end if they've actually done all that data analysis correctly. And then we can do that scaffolded communication with a poster or a manuscript. And because they have already done all the work and the bits for that, it really is more a communication assessment rather than a data analysis or understanding. And then the peer collaboration, it really enhances teamwork and reflection when they are working together on projects. So that's all I have. I'm going to stop recording, and maybe we can talk a little bit about your experiences with cures.